everyone, and welcome to another episode of BioLogic Science News. Today, I bring you news of a study that comes straight out of science fiction. Seriously, it's the kind of stuff that you would only see in movies. Researchers at the Worcester Polytechnic Institute were able to use spinach, specifically the veins of the spinach leaves, to transport blood pumped by a heart. You might be thinking, okay, how is this a big deal? After all, spinach is just one of many plants with vascular tissue, and all the vascular tissue does is transport water and nutrient-rich fluids like phloem around the plant. It shouldn't be too big of a stretch to assume that they could also serve as the transport corridors for blood, right? Well, not necessarily. Plant veins just aren't subject to the pressures and complications that animal veins are. Plant veins move water, and the water is slowly pulled up through transpiration, from the roots through the leaves. As water evaporates off of the leaves, it pulls more water up. It's a slow, perpetual movement of water through the vascular tissue of the plant. Animal veins move blood, which is thicker and heavier than the water and the phloem that plants move. Furthermore, this blood is not steadily drawn through the vasculature. It's pumped by a heart. This pumping mechanism creates pressure against the walls of the veins and arteries. And in addition to the regular physical movements of the animal itself, the blood pressure requires the veins to be durable and structurally resilient. When biological engineers try to design and create artificial tissues in the lab, like heart tissue or liver tissue, they need to find a way to make sure that nutrients and oxygen can permeate through all of the tissue. Biological tissue is saturated with small capillaries and veins and arterioles, and all of these little shuttling corridors help to keep the tissue healthy by bringing nutrients and oxygen and removing waste and carbon dioxide. One of the biggest problems for tissue engineering involves the recreation of these tiny veins. Because this is so challenging, artificial tissues often suffer from localized necrosis. Where the tissue is poorly vascularized, it can't readily get nutrients, it can't readily get incoming oxygen, and it can't remove waste and carbon dioxide. The tissue will starve, or it'll asphyxiate, or it'll get carbon dioxide poisoning. In the end, the tissue will die, and you get the localized necrosis. But what the researchers found was that the spinach leaves not only mimicked the small-scale vasculature, but that it could actually be easily modified to transport pumped blood. In their paper, the researchers detail their methods. First, they soaked the spinach leaves in a solution that dissolved the plant cells, and this left behind nothing but the framework of cellulose, like a ghostly outline of the cell tissue that used to be there. The cellulose connective tissue is built around the cells of the spinach, and so when the rest of the material, like the rest of the proteins and the organelles and all that gunk is removed, when it's all swept out of the tissue, only the cellulose remains, and it keeps the shape of the leaf and its veins. It's kind of like how in an office building, when everybody leaves and they take all their stuff from their cubicles with them and all the art comes down off the walls and all the posters and flyers and everything get cleaned up, you still have this empty building. You still have these corridors, these hallways and office rooms, but there's just nothing there. But here's the thing, is you can use those hallways and corridors and office rooms and redecorate them and repurpose them. And that's exactly what these researchers did with this cellulose framework of the spinach tissue. The researchers dipped the cellulose framework into a solution of living human cells. And so you see, cellulose is really biocompatible with human tissue. It's really biocompatible with animal tissue in general. And because of this, the human cells attached to the cellulose and they grew around it, surrounding the veins and coating the entire leaf in a grisly kind of reddish slime. This reddish slime, of course, is just the accumulated layers of millions of these human cells grown onto the cellulose framework. These human cells reinforced the spinach veins, and they provided the structural consistency necessary to support flowing blood as it's pumped through the tissue. The researchers say that this technique has a groundbreaking potential. It can be used to recreate the tiny vasculature necessary to sustain healthy tissues. It can solve the biological engineering problem of poor vascularization. It can allow us to create artificial tissues that will enjoy full access to nutrients, water, and oxygen. Tissues that won't suffer from chronic necrosis. This kind of biotechnology can allow us to create better implants, better organs like hearts and livers and lungs and bladders that will be more effective replacements for natural organs. This technique is cheap and easy, which makes it ideal for practical applications. You hear about so many cool things happening in labs across the country, but you wonder why they don't really appear too often or too frequently in consumer products. And this is usually because somewhere in the development chain, some really expensive procedure or some really energy-intensive process and it just slows everything down, and it keeps the technology in the lab. But with this, it's cheap and it's easy and it's quick, and so the potential for this to be applied to consumer products is huge. The raw medical potential here is really exciting because the tech offers potential solutions for a lot of stuff, some of which include the problems that currently plague biological engineering.